Hello students, hi. I am going to take up another revision lecture, but today I am going to take up embryology. It will be the first lecture which I am going to take up today. Now let us first speak about what is embryology. The definition of embryology. We know that embryo, we are talking something about the embryo. Now let us talk about the definition of embryology. It is the branch of botany which deals with the course of events leading to the origin of embryo. For the development of the embryo, the course of events start with the development of micro and megasporangia. In a microsporangium, what is a microsporangium? You've seen the stamen? You remember? I've already told you, yes, it is a stamen. In a microsporangium, the upper part, the anther globes, they are called the microsporangia. Microspores are produced, and from microspores eventually a male gametophyte, and within the male gametophytes later male gametes are formed. Microsporangium, inside this microspores are produced. What is the second name for microspores, or what are microspores also called? They are also called pollen grains. So formation of pollen grains within the pollen sacs. Microsporangia are also called pollen sacs, within the pollen sacs. And these pollen grains later behave as gametophyte and produce the male gametes. For embryo formation, male gametes have been formed. What we require now? We require the female gametes. Similarly, in the megasporangium or the ovule is the megasporangium, ovule or the megasporangium, formation of megaspores takes place. And eventually the female gametophyte, or also called female gametophyte or megagametophyte or embryo sac. And within the female gametophyte or the embryo sac, after double fertilization, the embryo and endosperm is formed. The entire megasporangium ovule with its enclosed structures. What are the enclosed structures of ovule? You have the nucellus, that is the megasporangium, and it's covered by integuments. What are these integuments? Yes, they are the testa and the tegument. After double fertilization, the ovule becomes the seed for next generation. Now let us first start with microsporogenesis. What is microsporogenesis? Formation of microspores within the microsporangia is called microsporogenesis. And where do you find the formation of microspores? Yes, this is the stamen. You can see one single stamen. It is here, the filament, lower part, which is attached to the anthers by virtue of a connective. This whole thing is called a stamen. If you take a TS, you can see the four sporangia you can see, or the four pollen sacs you can see. The structure of stamen is shown on the right hand corner of the slide, rather on the left hand side of the slide, sorry. Now for studying this, we have to study the TS of mature anther, how it looks like. Because here it is mature anther, the section has been taken, and we are just going to find out how does the TS of mature anther looks like. A typical anther comprises of 4 elongated microsporangia. You can see it very much, visualize it. See, so if you take a TS, you will get a figure or a structure like this. And at maturity, the 2 sporangia of each side become confluent, they fuse, owing to the breaking of partition between them. So actually it is TS of mature anther. We also call it TS of mature anther, quadrisporangiate sporangia or tetrasporangiate. Why? Because there are 4 sporangia. See, look here in this picture, the up and above the picture, where you can see the section of the stamen is shown. You can see the 4 sporangia. But at maturity, they fuse, 2 of them fuse, 2 of this side, 2 of this side fuse, and ultimately you can see only 2 sporangia. But actually they are 4 sporangia. What are the wall layers here? in a mature anther, because the anther, when it's mature, you can see the pollen grains, fully formed pollen grains with exine and intine, and you can see here the different wall layers. What are these? Epidermis, endothecium, middle wall layers, tapetum, the nutritive layer, and here is the connective. It is this very connective by virtue of which the anther lobes are attached to the filament, this pedicel-like structure or this Dundee-like structure. Look here, students, you have to remember certain things in the beginning only, because now we will be talking in terms of the cell divisions, the plane, the mode in which they are dividing. So what are the planes of cell division? One is periclinal and another is anticlinal. See here in periclinal, it is parallel to the surface of the cell, and here it is perpendicular to the surface of the cell. Can you see? Here it is perpendicular in anticlinal, in periclinal it is just parallel to the surface. So these are the two planes of cell division. And one more terminology we'll be using often in this particular slide, and that is in this study, that is cytokinesis of dividing pollen mother cells takes place either of the two methods. By either of the two methods, it takes place. You know what is cytokinesis? If it is mitosis or it is meiosis. The cell division comprises of two divisions basically. The first division is called karyokinesis. Karyon, the nucleus. First is karyokinesis, means the division of the nucleus. Division of the nucleus or karyokinesis is always followed by cytokinesis. Cytokinesis means the division of the cell. In plants, it is cell plate formation, and in animals, it is cell groove formation. So whatever the case, cytokinesis takes place after karyokinesis. So from one single cell in mitosis, you get two cells, whereas in meiosis, you get four cells. Now this division takes place in two ways. One is called successive division and another is called simultaneous. In successive, what happens as a result of meiosis? Meiosis 1, which is reduction division, the number of chromosomes is halved and two cells are formed. Two nuclei are formed and they are immediately separated by the cytokinesis into two cells. And these two cells in turn, after equational division, undergo another division and give simple mitosis. They give rise to four cells. So from one single cell, you get four cells in meiosis. Whereas in simultaneous method, what happens? The first division, the reduction division of meiosis, results in the formation of two nuclei. The chromosome number is half that of the parent. It is not followed by cytokinesis or cell plate formation. These two nuclei on their own undergo equational division and as a result produce four nuclei. Then after this, cytokinesis takes place and four cells are formed. You can see, you can visualize in this diagram, the four cells are formed. Thank you. Only this much in the present video, and please watch the second part. If you like it, share it and subscribe it. Thank you.